shape in time. So let me introduce the second speaker, Dr. Michelle Aizenberg. I don't think she needs much introduction to our field. So she's currently the professor of neurosurgery at our center. She completed, again, fellowship trained in neurosurgery, neurosurgical oncology at MD Anderson. She has been very active in leading research in neurosurgery field, and she's currently the PI on one of the investigator-initiated trial, collaborating with us on brain mets. And a lot of interest from her on imaging studies and collaborating with us on this primary GBM culture, which has started. So she has a very interesting title of the talk. I'm really looking forward to it. Without further ado, let's listen to how gliomas make friends and take over the world. All right, Michelle, we couldn't hear you. Probably muted yourself.
Sorry, I clicked it twice by accident. I said, thank you for the introduction. Let's see if I can get my screen sharing going. Are you seeing my slideshow properly?
We are now, yes.
Okay, great. So as Dr. Zhang stated, I'm going to talk about and explain how gliomas make friends and destroy the world. I do not have any relevant disclosures for this presentation. As a way of overview, we'll look at how gliomas overextend their welcome, and then we'll investigate how gliomas persuade and manipulate their neighbors, and then look at how we can assess functioning and the state of the brain. And then we'll look at how we can determine the effects of glioma sabotage and go over some conclusions. So first, let's look at how gliomas overextend their welcome. They do this by something called tumor microtubes. These are long cellular processes of the glioma cells, and their growth is driven by pathways involved in neurodevelopment and neuroplasticity. They are crucial for invasion and proliferation of the glioma cells, and they actually connect single tumor cells to a functional communicating network. And I'll show you a little bit more about that in a couple minutes. It has become clear that TMs, or tumor microtubes, are involved in neuron-glioma interactions. And what you see here in this color panel is blood vessels and tumor cells and the microtube extensions that are coming from them. And in this slide here, so this is day 40 and this is day 60. At day 20, you can actually see extensions into the, quote, normal surrounding brain. This is an EM showing two axons and their relationship with a tumor microtube. So tumor microtubes are not the same in all gliomas. When these investigators were looking at the length of tumor microtubes, they found that there was a higher density of shorter TMs in oligos and in lower grade astrocytomas. And as the length increased, it became clear that the higher grade tumors and GBM had the longest length tumor microtubes. This is just an illustration showing anatomical and molecular mechanisms of tumor microtube driven dissemination and network function. We have invasion, the extensions of the microtubes, which leads to proliferation and dissemination, and then interconnection with neurons forming in a network, gliomal neuronal network. And then the communication, this is a blow up of a communication between neurons and gliomas, which is facilitated with connexin 43 gap junctions. This is a little movie showing a xenograft stem cell mouse model, GBM stem cell, which is illustrating extension of ultra long processes at the invasive front of the tumor. And we're also going to see a single cell that has a protrusion, which is showing that scanning feature in that box. And how it's extending and retracting. This is a movie showing 3 different mouse models, which is going to illustrate the 3D morphology of the microtubes and the microtube interconnected cellular networks. You can see how dynamic it is and how the glioma cells are developing their processes, which are the tumor microtubes, and extending out, and can still see that scanning process. And this is the second movie showing another model. And then this last one is a genetic mouse model. And although it looks a little different, you can still see all of those same features with the tumor microtubes and the stem cells. So why are tumor microtubes so important? Well, they aid in resistance to therapy. And I'm going to show you how they do that. So this is a time series of a tumor region after laser-induced killing of a GBM stem cell, which is in this circle. So as the hours go by, we see that that cell dies. But if you look here, there's a tumor microtube extension that goes right to that area where the cell was killed. And there's translocation of a nucleus to that same spot. So I think this is pretty amazing. I mean, it helps explain why these are so difficult to treat. And so 60 hours later, you have this nucleus here, and then you're not seeing the tumor microtube. This is illustrating an area in the rectangle where there was laser-induced photo damage. And 2 days later, you see all of these connections and cells here, and the extent of the tumor microtubes that have facilitated this process into the photo damaged region. This are tumor microregions showing that at the start of radiation, and then 7 days later, there has been a change in the network. Before radiation, there were 11 cells that were not connected via tumor microtubes, and there's one exemplary cell here. And 7 days later, you can see the extent of TM formation that has taken place, and now there are only 4 cells left that are not connected. This is a 42-day time course of a distinct tumor microregion, and this is the start or right before radiation, and then going up to 42 days. And if you look at these cells with the asterisk, they survive the treatment. And the reason they do that is because they're connected with tumor microtubes. And if you look across the time period, you can see the change that occurs in these cells with the outgrowth of their tumor microtubes. So tumor microtubes seem to be crucial to glioma invasion, proliferation, and survival. What are they doing, and how do gliomas persuade and manipulate their neighbors? They do this with the help of neurogliomal synapses. These are direct synaptic communications between neurons and glioma cells, and they are functional chemical synapses. They are mediated by glutamate AMPA subtype receptors and may also involve NMDA receptors. And it has been shown that co-culture of glioma cells with neurons actually markedly increases proliferation of glioma. And we see that here. In this middle picture, there is co-culture of a neuron and glioma cells. The red are the tumor cells, and then the white is just representative of Ki67. When NBQX is added, which is an AMPA receptor antagonist, you can see that there's a reduction in the proliferation. But this reduction is not as great as is seen here, where the neuron is not present. So you can see that the presence of the neuron and this interaction between the two helps the tumor survive. This is just a close-up showing the neuronal and gliomal network and the communications. This is a blow-up of the synapse, showing the fairly typical process of communication, which then activates the glioma network, and then further increasing proliferation and invasion. This is illustrating via immuno-EM images, synapses with a low-grade glioma and also GBM. And you can see they both have this neurogliomal synapse. The brown is representing the presynaptic axon from the neuron, and the blue is the tumor microtube from the glioma. Here's a true synaptic cleft, and then there's also illustration of the docked vesicles at the synaptic cleft. So this communication results in hyperexcitability in the glioma, but the glioma cells induce hyperexcitability and seizures. And this occurs through secretion of glutamate and other synaptogenic factors. Interactions between neurons and gliomas are bidirectional and reciprocal. So they are both, so the neuronal network is stimulating the glioma network and vice versa. This is a study where investigators were looking at high gamma frequency power, and high gamma frequency power is correlated with neuronal firing rate and with local field potentials. And the high gamma frequency power is elevated by cortical hyperexcitability. And what they saw was that there was markedly increased high gamma frequency power in tumor infiltrated regions compared to the tumor core and to healthy appearing brain. So what kind of effect does this neuronal gliomal network and excitability have on brain function, and how might we be able to assess this? Brain connectivity may be a way to do this. So we're going to move on and talk about brain connectivity. Functional connectivity is what we're going to review first. Intrinsic brain activity has spontaneous fluctuations in bold signal. Bold signal, for those of you that don't know, is blood oxygen level dependent signal. And you can see here on the right that it looks like this. And it's way beyond the scope of this very brief talk to talk about this, but there's just a little tiny teaser. So resting state functional MRI evaluates the interactions that occur in a resting state when the patient is just laying there, not doing any task, utilizing bold signal. Functional connectivity measures temporal correlation of bold signal among spatially distributed brain regions. It assumes that regions with correlated activity from functional networks. This is a study that looked at functional connectivity, and they developed an abnormality or created an abnormality index by their processing. And without going into all that, what they saw was some very interesting differences between tumors. So on the left side here, there are 3 different glioblastoma patients. There's a large basal ganglia area one, a small left occipital, and then a fairly innocuous looking one in the posterior lateral right frontal area. But interestingly enough, all 3 of these patients have widespread, fairly global abnormalities in their functional network that you can see here. In contrast to that, in the oligodendroglioma patients that are illustrated here, despite the size of the tumor or the location, there's really more localized abnormality in the functional network as opposed to the global disruption that you see with glioblastoma. So structural connectivity studies the physical connections between brain regions by analyzing diffusion MRI. And it measures the movement of water molecules in the brain. And then tractograms can be reconstructed to capture the pattern and the trajectory of the actual connecting white matter tracts, which connect different brain regions. So what we then do is the cortex is parcellated based upon regions of interest, or ROIs. And those translate into these little spheres here, which are called nodes. The nodes are predefined collections of brain tissue, in essence. And then we can measure the functional connectivity between pairs of nodes by looking at the edges. And the edges are the lines that are connecting the nodes. And the weight of the edge or this line represents the strength of the connection. So how do tumor microtubes, neuroglioma synapses, and brain connectivity relate, and how do they affect the brain? Well, that is what I want to try to find out more about. So I have some thoughts, and one of them is that tumors that have a greater neuronal gliomal network will be associated with greater alterations in neurocognition and brain connectivity measures because of the increased tumor infiltration, proliferation, and neuronal excitability. And I also think that brain connectivity can mediate the relationship between neuronal gliomal interactions and neurocognitive status, and that connectivity changes will help us elucidate the relationship between high-grade glioma growth and neurocognitive status. There are currently and have not been any studies looking at brain connectivity longitudinally in relation to glioma neurobiology and neurocognition. And I believe that this may be a novel approach to detect recurrence of high-grade glioma much earlier than our current standard of care. And this may impact prognoses. So the aims of my proposed study are to measure the longitudinal effect of neuronal gliomal interactions on brain connectivity, and to also ascertain the sensitivity of brain connectivity measures to capture meaningful changes in cognitive performance and quality of life in newly diagnosed high-grade glioma patients throughout their treatment and through recurrence. And this will allow us to see what is actually happening as a result of tumor behavior and treatment. Is this actually feasible? Well, I believe it is, and there's some, some things that help support this. So first off, we know glioma cells can be found outside of the defined tumor margin that is seen on imaging or MRIs. Additionally, glioma cells have been found in brain areas that are absent of tumor as seen on histology. So pathology looks at the slide, says there's no tumor here, but when it's analyzed in other ways, other methods, there are actually tumor cells that are present there. Glioma cells also infiltrate along white matter tracts, and their highest density is peripherally. So these facts are telling us what we already sort of know, is that the tumor cell extension is much greater than just what we see on the imaging. So also things that support these ideas. Cognitive decline has been shown to precede MRI-defined tumor recurrence. So basically, the neurocognitive performance of patients declines sooner than the visible tumor recurrence on the scan. In gliomas, brain connectivity has been shown to be associated with tumor growth, patient survival, neurocognitive measures, treatment, and gene expression. And then lastly, we also have some pilot data indicating that our methodology is feasible and that we can detect correlations. So we have a pilot study where we're evaluating patients immediately pre-op and 2 weeks after surgery, looking at functional and structural connectivity and neurocognitive evaluations. Thus far, we've analyzed data for 12 of the patients. And this is an example of the processing for a glioblastoma patient. Pre-resection, we have the tractography here, and then we see the structural connectome and the functional connectome. And then post-operatively, we see the tractography again, and we see the structural connectome and the functional connectome. And you can see they look different. But what we really want to know is what's changing. And here, it's not the most robust colors, but the blue lines or blue edges are indicating connections that were lost after surgery for the structural connectome. And then this is for the functional connectome. And the red edges are connections that were gained after surgery. So this is illustrating that our processing works. We also are seeing some correlations between the connectivity measures and the neuropsych measures as well. So in A here, we're seeing that with an increased functional clustering coefficient, there is worsening in overall cognitive function. In B here, we see that increasing functional global efficiency results in decreased executive functioning. And then lastly, in C here, we see that increasing structural local efficiency is resulting in increasing hand dexterity. So for the molecular component of my study, I have not done any genomic studies yet, but looking at IHC in these glioblastoma specimens, we can see that the antibodies not only work, but that we're seeing some differential staining as well. So in conclusion, gliomas are aggressive because of the strong connection and communication that exists between glioma cells and neurons. And I hope I've also shown you that this also contributes to treatment resistance. The interactions of the glioma cells and neurons can result in hyperexcitability in the brain and seizures. It is not known how neurogliomal interactions affect brain function, but brain connectivity may be a way to assess this. Gliomas and their treatments affect brain connectivity, but currently in undetermined ways. Brain connectivity may be a way to determine glioma effects on brain function and serve as a biomarker for tumor behavior and tumor recurrence. I'd just like to acknowledge the Great Plains IDEA network for my funding my pilot study, and also David Ellis, my research associates, and my collaborators, neuropsychology, neuropathology, and neurology. Thank you for your attention.